Welcome to Study Loop.
All right, so we're in chapter 9, problem 10. And this one is all about the phosphorylation of a tyrosine kinase receptor, right? So if we imagine here, let this sort of be the plasma membrane. So I'll just do PM for plasma membrane here. If we have two of these tyrosine kinase receptors, right? So we'll have one and then two. Okay, and then here on the inside is going to be our cytoplasm, right? So that's the inside of the cell. Okay, and so we know that insulin will bind to one of these receptors. So insulin will come in here. So this is our insulin, right? And insulin will bind here too. And what happens is the first thing that's going to happen is that a dimer will be formed, right? So actually one of these is going to they'll sort of come together. These will, and so instead of having this distance, they're going to be nice little neighbors, right? So this way they are right next to each other, okay? Because these future steps here, we have the tyrosine residues in here and on the interior. These are going to be phosphorylated now, right? So the next thing that happens is the phosphorylation. Okay, so then now they're going to be phosphorylated. All right, and then the sort of last step is the cellular response. Right, so if we look at what happens here in the tyrosine kinase receptor, well, the first step one was to bind. Step two was this movement here. This was to form the dimer. Was step two, right? Step one being binding over here. 3 was the phosphorylation, and the last thing that happened was sort of the cell response. So when we're asked what happens, after the receptor has received insulin. So what happens after receptor, but before phosphorylation? Well, that's the step 2 here. It's the forming of a dimer. So the correct answer here is C.